Okay, this chapter is going to cover the information in the immune system, and I think a good way to start out is just to watch this animation that kind of gets you a good introduction into what we're going to be discussing.
This patient's cold is caused by rhinoviruses, shown here in red, that entered her nasal passages and escaped the first-line innate defenses such as mucus and cilia. Some of these rhinoviruses entered the respiratory epithelial cells where they replicated, releasing more rhinoviruses into the patient's nasal passages and tissue fluids. In the tissue fluids, cells of the second line of innate defense, such as this dendritic cell, carry out surveillance for anything that appears foreign. Dendritic cells engulf the rhinovirus within an endosome. The endosome fuses with a lysosome, whose enzymes digest the rhinovirus. The resulting rhinovirus fragments are called epitopes. The dendritic cell then attaches the rhinovirus epitopes to presentation molecules called MHC2. This complex folds outward so that it sits on the outside of the dendritic cell membrane. The dendritic cell then enters a lymphatic vessel and travels to a nearby lymph node. Here, the dendritic cell, part of the innate defenses, encounters a helper T cell, which is part of the adaptive defenses. The T cell receptor, or TCR, of this helper T cell recognizes the rhinovirus epitope, while the CD4 glycoprotein associated with the TCR recognizes the MHC2. The dendritic cell then stimulates activation of this helper T cell. Adaptive defenses help to control the current infection, but also provide immunity against future infections by the same rhinovirus. They are carried out by two types of lymphocytes, B cells and T cells. Immature B cells conduct surveillance in the lymph nodes, using B cell receptors, or BCR, to recognize only one specific epitope. The BCR on the left does not recognize the rhinovirus, but the BCR on the right does, and this B cell is selected for activation in a step called clonal selection. The selected B cell takes in the rhinovirus, degrades it, and presents the rhinovirus epitope with its MHC2. Our mature helper T cell recognizes the rhinovirus epitope with its TCR and the MHC2 with its CD4. Then, the helper T cell secretes chemical messengers known as cytokines that activate the B cell. The B cell proliferates and differentiates in a process called clonal expansion, resulting in two types of cell. Memory B cells carry the memory of this pathogen for many years. Plasma cells secrete antibodies that conduct surveillance for more rhinoviruses, grouping them together so they are easy targets for phagocytes. Antibodies in the blood and tissue fluids seek out free rhinoviruses. The antibodies neutralize the rhinovirus by binding to its surface molecules, thus preventing attachment to host cells. Antibodies are humoral defenses that target free pathogens. But what about the rhinoviruses that are already inside our patient's cells? Another type of adaptive defense cell, called a cytotoxic T cell, can locate and destroy rhinovirus-infected cells. It provides cell-mediated, or cellular, immunity. Let's return to our patient's lymph node to see how cytotoxic T cells become activated. The T cell receptor, or TCR, of this particular cytotoxic T cell is genetically programmed to recognize the rhinovirus epitope when presented by the dendritic cell. This time, the epitope is presented differently, with an MHC1, which is recognized by a CD8 glycoprotein on the cytotoxic T cell. With the help of cytokines from a nearby helper T cell, the
Okay, I guess it's going to stop there. So hopefully that gave you a little introduction to some of the terminology that we're going to be hearing as we talk about the immune system that we have. And you can see from that video that our immune system is very, very complex. There's a lot happening. There's a lot of adaptations and evolution that has occurred in order to make it so that we don't immediately succumb to viruses and bacteria and other things that get into our body. So a pathogen is an agent that causes a disease. So pathogens are often called disease-causing agents. And they can infect a wide range of animals, including humans. So anything that causes a disease would be a bacteria. It could be a parasite. It could be a virus. Those would all be considered pathogens. And the immune system that we have is what enables us to avoid or limit the infections that we get from those pathogens. We have animals, mammals in particular, but all animals have something called an innate immunity, which is a defense that acts immediately upon an infection. Vertebrates are very special because we also have something called adaptive immunity. So innate immunity would include barriers of defense. It's our nose, it's the hairs in our nose, it's our skin, it's mucus in our mouth and in our nose and on our eyes to try to prevent things from getting in. So it's a physical barrier. A small set of receptor proteins bind molecules or structures common to viruses, bacteria, or other microbes, and by binding to them, it activates a defense response to a broad range of pathogens. So all the things that our body isn't bombarded with, we really get sick from very, very few things considering what our body takes in because that initial innate immunity stops a lot of things. Now, adaptive immunity is also called acquired immunity, and this is what happens after we get exposed to something. So a great example of adaptive immunity would be what happens when you get the flu and you won't get that type of flu again. You become immune, or you get the chickenpox. You won't get the chickenpox again. You now become immune to that specific virus. So innate immunity is found in all animals and also in plants because plants have kind of a waxy barrier that prevents things from getting in. But in innate immunity in vertebrates is an immediate response to infections that serves as the foundation to that adaptive immunity. So if you look at other things other than vertebrates, insects have that exoskeleton. Our digestive system is something that is considered an innate immunity. It's got a lot of stomach acid in there, so when we consume food that isn't at its prime, hopefully our stomach acid will help break that down. If we consume something, you bite your fingernails and you're consuming and putting into your body bacteria and viruses and all sorts of other pathogens, hopefully your stomach will kind of break that down for us. We have this enzyme within ourselves called lysosomes, right? And those things will help. We have types of cells called lysosomes, and within those is an enzyme called a lysozyme, and it breaks down bacteria walls and just kind of eats away and kills it. Immune cells of insects recognize pathogens by binding molecules specific to the virus, and each recognition protein of insects recognizes a broad class of pathogens. Hemocytes circulate within the hemolymph of the insect, and they carry out that phagocytosis, where it engulfs and it eats and ingests whatever the bad thing is in the body. So even simple organisms like an insect contain certain things as a part of their immune system that help them, much like humans do. So here we have lysosomes that contain that enzyme lysozyme, and if this is a bad thing, well, first of all, pathogen comes in. These little purple things are pathogens. The let's see. These little pathogens come in contact with the outside of the cell. The cell will engulf it and form a little vacuole that has the bad thing inside of it, like you see here. The lysosomes attach to it, dissolve it, and then spit it back out. It's useless now. Can't do anything bad, hopefully, by the time all of that has occurred. The immune system of mammals is the best understood of the vertebrates, mainly probably because it is the most often studied. And so our innate defenses include barrier defenses like our skin, phagocytosis like we just looked at, and then antimicrobial peptides. Additional defenses that are unique to vertebrates would be natural killer cells, these things called interferons that we'll talk about a little bit, and then the inflammatory response. So that barrier defense is going to be skin, our mucous membrane of our respiratory system. We have a very moist respiratory system, right? So when you breathe in, your throat is wet. There's cilia in there. There's other things. The urinary system, urinary tract is also a type of barrier defense, kicking things out that don't need to be there. Reproductive tracts are also a barrier defense. Mucus really grabs a lot of stuff. You have to blow your nose all the time when you're around gunky things, right? When you're sick, you have a lot of mucus that builds up. That mucus is taking junk out of your body for you. So that's also usually a sign when you have a lot of mucus that you've got something going on. Many body fluids like our spit, our saliva, mucus, tears are pretty hostile to a lot of microbes. Their tears are super, super salty. Mucus will have a little bit of stomach acid in it, as well as the pH is going to be pretty high. Saliva is the same thing. So also our skin has a fairly low pH. Our digestive system certainly has a really low pH, meaning it's very, very acidic. And so a lot of bacteria can't grow. It comes in, but it can't continue to grow. And so what little bit is in our body's able to get rid of on its own. Pathogens that enter the mammalian body are subject to phagocytosis, where those cells recognize that that's not something that should be there, and they engulf and take them in. The lysosomes attach to that little vacuole that was formed, and it dissolves it and spits it back out of those cells. There are two types of phagocytic cells. So neutrophils circulate around in the blood, and they are attracted and then become activated and move and stay put when there are signals from infected tissues. Macrophages are just found everywhere. So a neutrophil circulates throughout the blood. A macrophage is found throughout the body. Other types of cells that I would be familiar with that play a role in innate defenses. There's dendritic cells that stimulate the development of adaptive immunity in cells that connect the environment, such as skin. So we'll talk about what adaptive immunity is in just a minute. Eosinophils discharge destructive enzymes which will dissolve, eat up, destroy a pathogen that's in the body. We also have, so vertebrates have these things called natural killer cells. These aren't going to be found in non-vertebrate type things. And these just circulate through the body. They pick up and detect when there's something abnormally happening, and they're attracted to it. And when they are attracted to it, they release these chemicals that lead to cell death, killing a virus cell or a bacterial cell or something like that. But they also inhibit the spread of virally infected or cancerous cells. So natural killer cells are really important when they're really strong and going well. In our bodies, there's oftentimes when we have cancerous cells, things that are growing out of control would be the definition of that. And these natural killer cells help reduce the rate of that growth. In mammals, pathogen recognition triggers the release of these things called peptides and proteins, and those things will attack the pathogens or at least impede their reproduction. So at least they're not going to make more of themselves and grow. Interferons provide an innate defense, so they interfere with viruses and they help activate those macrophages, which are those cells that are important in getting rid of bad things in our body. And the complement system consists of about 30 proteins that are activated by substances on that microbe surface. So activation can lead to lysis of invading cells. Lyse means to slice or to split open, so it's going to bust those invading cells. We also have this thing called an inflammatory response, and that's where you get a paper cut on your finger, and it gets red and it gets hot and it doesn't feel well. So pain and swelling is an inflammatory response. So around a place of injury or infection, it's brought on by these molecules that are around it. So while it's painful to us, it's also an indication that you have an infection going on inside of your body, or at least at that immediate location. So when you have pain and swelling, generally, if it's really, really bad, your whole arm swells up, something like that, antibiotics are important to reduce that infection. Activated macrophages and neutrophils, remember those things that are floating around in our body, release something called a cytokine signaling molecule that modulates the immune response and promotes blood flow to the site of injury or infection. So these macrophages release a certain enzyme. It tells those molecules we need more blood flow into that area. You know, when you get infection, the blood is pounding in that area. That's where the heat comes from. That's where the red comes from. That's why it's a little bit more swollen also. And then mast cells will release a histamine, which triggers the blood vessels to dilate and become more permeable so that they are able to take up the bad stuff and try to get it out, like usually like through the urinary tract infection, somehow like that, and also to release good things like white blood cells to try to fight that infection. So that increased blood flow that results to the site helps deliver antimicrobial peptides as well. And so you usually will get an accumulation of pus, which is fluid-rich white blood cells. And it's also just other junk like debris and dead pathogens and damaged tissues. It's just trash. And either the body will reabsorb it or it grows so large that you have to have it lysed or something like that. But basically, that's what pus is. It's an accumulation of white blood cells and junk. It's an indication that there was an infection at that initial site. So this is an image of that that inflammatory response that we see. So we have some sort of a pathogen like a splinter. The splinter maybe is removed, but it has all this bacteria that's attached to it that it leaves behind, right? All right here, and it left a bunch behind over here. So immediately when that splinter is there, you have mast cells that release these signaling molecules, macrophages that are going to try to engulf what kind of breaks off. The red blood cells in the capillaries, right, become a little bit more permeable. So these peptides now can leave the body and go eat away at that junk that's there. And then lastly, phagocytosis, where it's going to eat away at what is left behind. So inflammation can either be local or systemic. If you're lucky, it's local, right? A local would mean that it's just at the site where it hurts. Fever is a systemic inflammatory response. So that means, you know, you ingest something, you get a fever, you have a cold, or you have the flu. Your whole body is responding to this pathogen that's inside of you. When it gets really bad is when we go into sepsis or septic shock, which is life-threatening, and it's an overwhelming inflammatory response. So your organs basically just shut down. Chronic inflammation can also threaten human health as well. So a lot of adaptations have occurred to and evolved in some pathogens that enable them to avoid destruction by the phagocytic cells. So just as we have evolved all these amazing ways to fight these bad things, those bad things have evolved amazing ways to counterbalance that. And I guess an example would be tuberculosis resists the breakdown within those lysosomes, so that enzyme lysozyme can't break it down after it's been engulfed by the host cell. So the adaptive response relies on two types of lymphadocytes or white blood cells. So we just talked about innate immunity, all these things that our body has that prevents us from getting sick. Well, the adaptive immunity, remember, is something we have to be exposed to something first usually in order to gain this adaptive immunity. It's a little bit slower acting than innate immunity is. And it's also only found in vertebrates. So lymphocytes or white blood cells, that mature in the thymus above the heart are called T cells, and those that mature in the bone marrow are called our B cells. So these are the two types of lymphocytes. One is made above the heart and one is made in the bone marrow, T cell and B cell. Antigens are substances that can elicit a response from a B or a T cell. So when recognition occurs when a B or T cell binds to an antigen, it's called an antigen receptor. Or an antigen receptor is sticking out of these cells, right? Binds to a B or a T cell, and then recognition occurs. Now an immune system will start to happen. An immune response will start to happen. You get millions of different antigen receptors, but the receptors on a single B or T cell are all identical to one another. A small accessible part of the antigen that binds to the antigen receptor is called the epitope. In your book, there should be a really good image explaining what all of these things look like, and now would be a good time to pop that open and take a look at that. Each B cell antigen receptor is Y-shaped. The constant C regions of the chain vary a little, whereas the V regions vary greatly. So you have, a Y shape, and then the down part of the Y would be called the chain. Those V regions vary because they're going to bind to certain things specifically. So here's one of those pretty good images. This whole B cell is our lymphocyte, right? Our white blood cell. And on the outer part of it are these things called the antigen receptors. And the Y part of it, or I guess the V part of it, can vary greatly, right? This part's the chain, and they're going to be pretty similar among the different ones. But there's many, many on a lymphocyte so that it can bind to different pathogens correctly. When something binds to that antigen receptor, that's the first step. They call it the activation. And in this case, it was a B cell instead of a T cell, so you get activation. That makes the cell start to secrete out an antibody or immunoglobulin. Secreted antibodies are similar to B cell receptors, but they're not membrane bound, so they're just kind of floating around. So the antibodies, rather than the B cells, actually defend against pathogens. B cells release something. There is a receptor that says, "Ooh, this isn't good. Something shouldn't be here." That makes the B cells start to release the antibody. The antibody floats around, and it starts to defend against the pathogens.
The main role of antibodies secreted by plasma cells is to tag foreign molecules or microbes for destruction. Binding of antibodies to antigens can trigger several effector mechanisms. Neutralization occurs when antibodies bind to antigens, blocking their activity and making them harmless. For example, antibodies cover the surface proteins of this virus, preventing it from hooking up with the host cell. Other antibodies may bind to toxin molecules on a bacterium. The bound antibodies attract macrophages, which dispose of the invaders by engulfing them, a process known as phagocytosis. Antibody molecules have at least two binding sites, allowing them to form a clump of microbes by binding to two antigens on two different cells. This process, called agglutination, traps microbes and makes it easy for macrophages to capture them and dispose of them by phagocytosis. Precipitation is similar to agglutination, except this process clumps together free antigen molecules dissolved in body fluids. This inactivates them and attracts macrophages, which destroy them by phagocytosis. One of the most important mechanisms of humoral immunity is the activation of complement proteins. Binding of antibodies to a foreign cell triggers complement proteins in body fluids to form a hole in the membrane of the target cell. The hole allows water and solutes to enter the cell. The foreign cell swells, bursts, and dies. Humoral immunity, mission accomplished.
Okay, so this is just a summary of basically what I talked about, right? The B cells bind to something, this pathogen here, and that causes the antibodies to be released, right? And they look a lot like these receptors, but they're not actually receptors. These things, these antibodies floating around, are going to attach to the antigens and ruin the pathogen that's in there. So our adaptive immune system has 4 major characteristics. We have those lymphocytes, and we have the receptors, and they're very diverse. We have something called self-tolerance, so the lack of reactivity against an animal's own molecules. So we shouldn't, those antigens that come out should not fight our own body, shouldn't like attach to our liver and ruin our liver, right? They're real specific. Proliferation of B and T cells after activation, they start making a lot more of themselves. And we have this thing called an immunological memory. So after you react to something, you will likely react to it again, like in the case of allergies. Also, after you have already had a virus, your body remembers that and it's able to knock it out. So it's not going to be able to give you the flu a second time if it's the same specific type of flu. You're not going to be able to get chickenpox or measles or something like that a second time unless the virus is going to change itself. Antigen receptors are generated by random rearrangement of DNA. I'm going to skip through that because that's not really something I want to get into much depth with. What I would know here is that, so memory cells give rise to effector cells. If the same antigen is encountered again, I would know what a memory cell is. Effector cells are essentially just cells that are like short-lived. They're activated for a short period of time, and they act real fast immediately against that antigen. So memory cells will make more of those real quick effector cells if the same antigen is encountered again, and that is how we get that immunological memory.
Humoral immunity, carried out mainly by B cells, deals with invaders in body fluids. B cells are lymphocytes that send out antibody molecules in response to certain stimuli. There are many kinds of B cells. Each kind produces and presents its own uniquely shaped antibody receptors in its plasma membrane. These unique receptors only bind to antigens with specific shapes. The B cell that successfully binds to an antigen is stimulated to proliferate into a clone of cells with the same specificity. This process of antigen-selected proliferation is known as clonal selection. B cell proliferation produces clones of plasma cells and memory cells. Memory cells are long-lived cells that can respond rapidly to future exposures to the same antigen. They account for the speed and strength of the secondary immune response that gives us long-term immunity to a disease. Plasma cells multiply rapidly. They produce and secrete antibodies in increasing amounts, peaking about 2 weeks after the activation of the original B cell. These antibodies flow through the body's fluids and tag the original foreign antigen molecules, marking them for destruction.
So that immunological memory is responsible for long-term protection against diseases due to a prior infection. So you had to be exposed first. The first exposure is called the primary immune response. And during that primary immune response, the B and the T cells give rise to their effector forms. Secondary immune response is a much quicker, faster, stronger, longer response than the primary one was. Immunological memory can span many, many decades. So another example would be like when you're given a live or a killed virus as a vaccination, what you're trying to do is cause a, like you're getting a primary immune response, and your secondary immune response would be that it's already in your body, right? And if you are exposed to chickenpox or whatever else might be the vaccination that you're getting, you don't actually have the secondary immune response. Okay, so those B and T lymphocytes produce a humoral immune response and a cell-mediated immune response. The humoral immune response is basically antibodies are helping neutralize and eliminate those toxins and pathogens in the blood and the lymph. And the cell-mediated immune response is where T cells destroy infected host cells. So humoral immune response, it's in the blood, it's in the lymph, it's just trying to get rid of it before it enters the cell. And in a cell-mediated immune response, these pathogens have already entered the cell, and so the T cell will actually destroy the cell. A type of T cell called a helper T cell triggers both that humoral and cell-mediated immune responses. To produce this response, a foreign molecule must be bound by the antigen receptor of the helper T cell. An antigen must be displayed on the surface of an antigen-presenting cell. I'm going to skip through this a little bit just to watch this video.
Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface along with a self protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activates specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.
Okay, I would also note, so antibodies don't kill pathogens. Instead, they just mark them for destruction. So that'd be a really good true or false question. Antibodies may also bind to toxins in body fluids and prevent them from entering body cells. So B cells can express 5 different forms or classes. One type is membrane bound. The others are soluble, so we get them. So there's B cells that are found in our blood, our tears, saliva, and in breast milk. That's why they say that it's so important for newborns to have a little bit of that breast milk. Now there's a lot of formulas, at least for humans, that can be effective, but it's especially for young mammals in the animal kingdom, it's important for that baby to nurse from the mother in order to gain better immunity. Cytotoxic T cells are the effector cells in the mediated immune response. Cytotoxic T cells recognize fragments of foreign proteins produced by infected cells.
Now let's look in more detail at cell-mediated immunity. Cytotoxic T cells are the main warriors of cell-mediated immunity, which attacks body cells that have been altered by infection or cancer, or transplanted cells that are foreign to the body. An infected, cancerous, or transplanted cell displays foreign antigens on its surface. T cell receptors on specific cytotoxic T cells recognize and bind to specific antigens on cells. Cytotoxic T cells respond to this contact and to interleukin-2 sent out by helper T cells by producing the protein perforin. Perforin molecules are released from the cytotoxic T cell and attack the membrane of the infected cell, punching holes in it, which allows water and solutes to enter. The cell bursts and is killed. In this way, cytotoxic T cells deprive pathogens of their host cells so they cannot reproduce. Tumor cells are also removed from the body by cytotoxic T cells. In this way, cell-mediated immunity, carried out by cytotoxic T cells, destroys altered or infected body cells.
Okay, so to kind of summarize everything, both the humoral and the cell-mediated responses can include primary and secondary immune responses. Memory cells enable the secondary response. Active immunity occurs naturally when a pathogen infects the body, and passive immunity provides immediate short-term protection. So I would know the difference between passive and active immunity. Both passive and active immunity can be induced artificially, and it can also be conferred naturally when antibodies cross the placenta from mother to fetus or pass from mother to infant during nursing. Active immunity is induced when antigens are introduced into the body in vaccines. So in this process of it's called immunization. Inactivated bacterial toxins or weakened or killed pathogens, so a live or a killed virus, but the virus will be weakened, are introduced. And passive immunity can be conferred artificially by injecting antibodies into a non-immune person. So although adaptive immunity protects against many pathogens, it's definitely not fail-safe. So one great example is allergies. Allergies are exaggerated responses to antigens called allergens. So when you get an allergy against hay fever, oak pollen, cedar in the wintertime, plasma cells secrete antibodies specific for antigens on the surface of the pollen grains. And this triggers the immune cells in the connective tissue to release histamine, which is an inflammatory type chemical. So the pollen is in your nose, it's in your sinuses, it's in your lungs, and you get this inflammatory response. So the nose gets clogged up, you're coughing, mucus is formed, that kind of thing. Antihistamines block receptors for histamine and diminish the allergy receptors. So when you take Benadryl or some other antihistamine, you're blocking those histamines, which are causing inflammation. So an allergy is like an exaggerated response to an allergen, causes a lot of inflammation, and as many of you know, a lot of pain. An acute allergic reaction response can lead to anaphylactic shock or a life-threatening reaction. So basically, certain things, you just have an acute would be the opposite of chronic, meaning it's very fast, very quick, intensive amounts of histamines are being released, and can cause shortness of breath. The lungs kind of shut down. The heart goes way too fast or way too slow. A lot of these things would be like different types of venom, not just bee venom, but wasps and fire ants here, especially in Texas, penicillin, peanut, shellfish. So people with these hypersensitivities often have this epinephrine pin, the EpiPen, which will counteract the allergic response, open things up, allow the heart to beat very, very quickly. Another problem with our immune system is when we have autoimmune diseases. So people that have an autoimmune disease, their immune system targets certain molecules of the body. And so we are reacting, acting against our own cells, basically. And there's a lot of different types. Multiple sclerosis is an example. Diabetes is another example. Type 1, not type 2. Lupus, rheumatoid arthritis, like you can see in that picture. And there's a lot of things that can influence these disorders or influence your susceptibility to them. Genetics, heredity, a virus that maybe you received that ended up causing something like this. All of those things influence your susceptibility. And then immunity plays a role in cancer. So the frequency of certain cancers will increase when your adaptive immunity is impaired, because about 20% of all of our human cancers involve a virus in some way. So the immune system can act as a defense against viruses that cause cancer and against the cancer cells that harbor the virus. So if you can recall the HPV vaccine, that's a virus that's associated with cervical cancer. So when people do use, when they get the HPV vaccination, that's a virus that males can give to females, females can give to males, and the males can then give to more females. While males may not react to this virus and we may not get sick from this virus, if we are exposed to it, it can cause cervical cancer. So getting the vaccination against this virus helps prevent cervical cancer.